Hello, everyone. I'm Jeff Agar, Associate Dean of MS Education and Lifelong Learning here in the College of Science. I'll be introducing the speakers. So I will start with Jared Auclair. Jared, after a PhD in WPI, went on to be trained by a very famous scientist, Greg Petsko at Harvard. And I've known Jared a long time. I knew him when he was only 6 foot tall. He's done a lot of amazing things that aren't on his resume, but one of those is the growth of BATL, and the thing that I find the most impressive, being able to somehow put together a CLIA-certified COVID testing facility that at one point, Northeastern was doing more than 6 or 7 states could do combined. So I'm very proud of that, and that just talks about Jared's effectiveness. Now, if you know Jared, you know to be afraid of him, so I want to read his titles from an email he recently sent, so I'm going to start there from the bottom. Agar, if you get this wrong, they'll be held up. Oh, that's the body. Let me get to this. So, Vice Provost, Research Economic Development, Director of Bioinnovation, Associate Teacher Professor, Chemistry, Adjunct PI, National Institutes of Bioprocessing Research and Training, NIBRT, Northeastern University Innovation Campus at Burlington, otherwise known as BATL. The BATL's on that campus. And now, we're proud to say, the Dean of another college, CPS. Jared, come on up.
Thanks, Jeff. I'm sorry, AV guys, I'm Roman. It's just my style. I was trained by Greg Petsko, who doesn't stand still, but obviously by Jeff. So congratulations to the Institute for being 50 years. I remember the 40th, which is when they recruited Jeff to come over to Northeastern from Brandeis, and I followed, and he was the good person they recruited, and I just never left, hence why I have all the colors. And to the Barnetts, congratulations, and thanks for the continued support. So I'll tell you a little bit of a story, but before I do that, I want to say Barry likes to give advice and opinions, yes? So I'm going to ask you to make your 3 M's 4. Manufacturing should be added. So you have manufacturing, molecule, measure, and meaning. What do we think?
Yeah, let's do 5 for money.
Ooh, money! Rick, did you bring the money?
I have before.
I know. So, this is just what I'm going to talk about, which is everything that you've heard about, so we won't belabor that. So here's a little timeline of the work I've done at Northeastern since I've been here in 2013 when we came over. I always tell people they sent me to run BATL on the Burlington campus as I was looking for a tenure-track faculty job because Jeff ran out of money to pay me, and they had to pay me somehow. So they sent me there, but the intent was that the whole thing would fall apart over time, which is not how I roll. So we continue to build that from 2014 until today. And these are the other things that I'll talk about in terms of our regulatory training with APEC, ICH, and the Life Sciences Testing Center. I do research also, I just don't only train. And a coalition that I started in 2020. But really my thing that I think is most interesting for me and why I continue to work here at the university is to take a page out of the NSF's book and their new directorate, the TIP directorate, and really focusing on technology, innovation, and partnership. And for me, that's at the intersection of workforce development, use-inspired research, application-based research, and entrepreneurship, and doing all of those things, or trying to. I'm going to steal just 10 seconds of thunder that we came out of Barnett, and we are also 10 years old, and in the last 10 years, we've grown in scale. Due to many of you here who've helped, Rohan, Wasim, and many others. Rick, although Thermo doesn't like me anymore since you left. I'm just kidding. But it's all good. And you'll follow on the timeline down there where I am. So BATL started then, and as many of you might know who've interacted with me over the years, I can't say no to anything, hence why I got stuck doing the COVID lab. And I'm all about seeing opportunities and trying to learn from Barry and identify what the next big thing will be to look at and to continue to work on. So we do focus on biologics, advanced therapies, vaccines, and a little bit of medical devices with CMC quality, AI and machine learning, and data integrity at its core. It's not only you, Bear. The slides are all messed up. So really, that's our focus in what we look at. The thing that's really interesting that I want to really emphasize when I'm talking is I got my bachelor's degree. I graduated and said, "Oh shit, I don't know how to do anything. What am I going to do next?" Default, I'm going to get a PhD. What else would I do? I finished my PhD and I had one set of skills, but not many. So I wanted to expand those and did a postdoc. All that to say that I knew nothing about any of these things. Literally almost nothing, right? If you talk to me about biotechnology, and many academics talk about biotechnology, and they don't know what it is still, right? And the alphabet soup beyond mass spec of CQAs, CPPs, and other things, I learned it all by doing essentially on the job, right? So it's never too late to try something new and to take a risk. So BATL, because I'm not going to say the words, there are too many. So BATL is really an opportunity to look at the entire life cycle of a molecule in a non-GMP space. Not as fancy as Jonathan set up. I lost him, there he is. But in a similar sort of thought process, right? So a third of the lab is CLIA CAP accredited. We'll talk about that in a little bit. A third of the lab is really focused on biomanufacturing and process analytical technology, integration, automation, and flexibility. And a third of the lab is really focused on analytical characterization. Historically, mass spectrometry, but beyond. So we do genomics, we do proteomics, we do metabolomics, and the instrument company that wants to test out a new instrument, we put them in the lab and we put them through their paces. And then we let you know that they're total crap and start over. No, we don't. But we do provide honest feedback. We do, don't we? Okay. But everything I do is in the lens of regulatory science, which is an interesting space to be in academia because it's not a space that many think about. In regulatory science in the sense of quality and chemistry, manufacturing, and control. So as David Leslie mentioned earlier, 2014, I went out to BATL, started training industry. Awesome. I can do this. It was great. It was a lot of work finding people to come to be trained, but we can do that. So then a bunch of stuff happened in the chemistry department, and in the summer of 2015, David came to Jeff and I and said, yeah, there's this APEC thing, the Asia Pacific Economic Corporation, that we're supposed to be involved in, but nobody's been answering the emails from the organization for 6 months. Is that something we want to do? And I'd already told you, I say no to nothing. So I said, yeah, let's do that. So we went. I remember we flew down to Peru. Back in the day, I didn't like to travel. Was not a good experience for me. Now I travel all the time, everywhere. We went there and became a center of regulatory excellence in biotherapeutics, where we started training regulators from the Asia-Pacific region how to do science-based, risk-based evaluation of drug product and drug substance. And we continue to do that today. We expanded into cell and gene therapies. We dabble in RNA and vaccines for them and other modalities as well. We also, I used to be a really quiet guy who would sit in the corner and want to not be seen. That changed someplace along the line and started speaking up at the meeting. And now Northeastern sits at the leadership table for these centers of excellence, driving the future of what they'll be and where they're going to go. And getting texts from people at the FDA who are asking me to do things that I don't get paid to do. And then I meet them and I'm like, how many houses do you have? But they go work at industry, so that's why. So we started training, and then through relationships with, as Barry mentioned, with SEAM and others, we started a relationship with ICH, who started a training program 2018. In 2018, where we started training regulators on ICH-Q1, drug stability. And then we started to do Q2, Q3, Q4, all the way up through Q14 now. We're expanding our footprint through hiring of some new staff and partnering with Harvard's MRCT and looking at some of the clinical aspects and really thinking about the entire interaction of the drug life cycle. And what's really interesting is we took a multi-prong approach to the training, two-prong approach. We fly people to me in Burlington and train them, including on how to, we put them in front of the instrumentation so they understand where the data came from that they're analyzing. Because the scariest thing is, regulators look at dossiers all day and data, but they oftentimes have no idea how the data is collected or how you might manipulate the data. So we spend a lot of time teaching them about those kind of things. And we continue to do that today. And it's not only the developing agencies, but even the US FDA, we usually use them as trainers. And at the end of a training, they'll say, oh, we didn't know that. It's been a long time since we've been in a classroom. So it's an opportunity to train, right? That was the original model. And then it started to be me and a small team of industry partners traveling the globe, training regulators, because it's more cost effective to send me and 3 people to a location than for me to pay for 30 people to come to me. So we started to do that. And as a matter of fact, I was in Kenya last week, training a group of African regulators in industry on quality and regulatory aspects of vaccines from through the Wellcome Trust. And it's always great to get a different perspective and try to help get drugs to patients. That's why I do a lot of these things. I also do this for for the benefit of students, people and patients. And everything we do is credentialed, experiential, and stacked. This is every place in the world. You can't see the globe, but you can see every place in the world we've trained in orange. I'm so crazy, my wife said, that I went to Kenya last week, and I was in the country for 18 hours. I slept in my bed on Monday night, and I didn't sleep in another bed until I got home on Friday night. Thanks to Heathrow's delays. We also expanded into the World Health Organization and partnering with a coalition of interested parties in regulatory system strengthening, where we are also helping shape what the CIP looks like as one of the 20 founding members and only academic institution at that time, to really help system strengthening in Africa, Latin America, Asia, and other locations, using the global benchmarking tool as a model, right? What is the thing that we are always looking for? KPIs. WHO gave us a tool that we can use as KPIs. Is our training helping a regulatory authority go from maturity level 2 to maturity level 3? And maturity level 3 is essentially a functioning regulatory authority. And just because I pay attention, the whole concept of stringent regulatory authorities is now being replaced by WHO-listed authorities, which are linked to these global benchmarking tools. But like I said, BATL doesn't only just do training. We do research. My philosophy on the lab is it's a collaborative space where faculty, industry, anyone can come and partner with us on research projects. Some of those I lead, some of those we partner with Jeff, with Sunny Zhao, with others. So it's just a smattering of a few things that we've done over the last few years, including one that was really fun here. Oh, you can't see it, right. Where we did an evaluation, we did an inspection of a manufacturing facility using mixed reality to see if that might be a tool that we can use in the future when travel is limited through things like a pandemic, but also because many regulatory authorities can't afford to travel and inspect. Plus, companies don't want to be inspected every other week by a new regulatory authority. At least that's what they tell me. I believe it. One of the spaces that we're going down and we're looking at aggressively now is some of the things that Jonathan talked about in terms of AI and machine learning. And I think about AI and machine learning in 2 buckets for me: process and product. How can we make better product using AI and ML tools? And I won't talk about that because Laurent's going to do a great panel this afternoon. So he'll mention some of those things that they're thinking about. Everything I do is in partnership. My goal is to surround myself with smarter people than me, except for Jeff. Yeah, I figured. And then process, things like Jonathan was talking about. One of the things that I'm really interested in is process analytical technology. The less a human touches the process, the better, because we will add problems to the process. The process is the product. And we partnered with, I can't even list all the partners, so I just threw up a few here. Many of our partners are listed here, Genentech, Biogen. We partner with many regulatory authorities, FDA and others, state organizations like the Mass Life Sciences Center, USP. We try to keep our finger on the pulse of what's happening. That's the goal. So as I was going along doing that great work, and I got a $4.25 million grant from the state of Massachusetts to expand the lab space, they handed me the keys to that new lab space in Burlington on February 29th of 2020. It was awesome. I did a training for 15 regulators from Africa on March 9th of 2020. That whole week I was praying that they weren't going to close down the university because I didn't know what I was going to do with these 15 people. They all literally got the last flights out of Logan back to the continent of Africa on March 13th before everything closed. Otherwise, my wife and I would have had house guests for a long time, which is fine, a long time. But because I had that empty lab space that nobody was using, and they went to David and said, who's going to do the COVID testing? And I was like, well, duh, of course I can do that. That's easy. We set up the CLIA CAP lab. He said usually it takes 6 months to get approval. It usually takes 18 months to get licensed, which we did in 6 weeks. We had 80 people working there at one point, of which July 14th, we had our first floor employees start, and we started testing on August 1st. We did 2 million tests over 2 years, 24/7, nonstop. We changed processes from pooling to non-pooling and a whole bunch of other things. The lab still operates. I absolutely refuse to close the lab. They will have to force me to do it. So it's about finding other uses. We can use a CLIA CAP lab to develop bioassays for the biotech industry because it mimics GLP. We can do clinical tests. I'm going to try to use it to leverage an opportunity to bring in actual GMP into the site. But the biggest thing that we do right now is in partnership with Ginkgo Bioworks, wastewater testing from airplanes, from hospitals, from several other random places where one collects wastewater so we can track pathogens. So we can see what is circulating and what we might need to worry about next. I'm also, I'm always about an opportunity. We got, the university, it was a great time, by the way. The university's like, what do you need, Jared? I'm like, this thing. Okay, here's $1 million. I'm like, awesome. I'm like, this thing, here's another $1 million. I'm like, great. That's long gone. But it was all reimbursed by the government, or most of it, and I leveraged that to upgrade our capabilities in the cell and gene therapy space and the genomic space to bring in new equipment. So, leverage those opportunities. But in the LSTC also, we just don't do testing. We're starting to do training, med tech training, clinical research training. We also published several papers on testing in higher education, on partnerships on new diagnostic models. Actually, diagnostics and medical record technologies are so old school, it's scary. There's lots of opportunity there. Success, when you do something right and well at a university, probably any place, then they come to you and say, can you do this other thing? It's going to be really easy. There's this Build Back Better Regional Challenge grant application from the CHIPS Act. It'll take you a half an hour, Jared, to write it. It's no problem. Yeah, that sucked my life up for 18 months, but was probably the most fun I've had in writing a grant and making it through phase 1 funding to build this coalition, BioConnects New England. And it's really about a regional approach to biotechnology. Massachusetts, if I asked you all if Massachusetts was the biotechnology hub of the world, you would resoundingly say nothing. You would resoundingly say, and I would say no. We're the biopharma hub of the world, but biotechnology is way more than drugs. It's way more than drugs. It's bioag, biofuel. It's about using living organisms to make a product. We do drugs really well, but there's other opportunities. So let's think about how we can take the research and development in Cambridge and Kendall Square and spread it across the rest of New England so we can remain competitive with states like North Carolina, with California and others. And these are just some of our partners. But it is based on what I believe in, workforce, research, and entrepreneurship to drive the coalition. In developing interesting models to get people interested in working in the life sciences. Many of us have had opportunities to enter the field. Many people in the US and around the world don't even know what a biotechnician does or is. Everybody knows what it means to be a medical doctor, but the key driver of the New England economy or the Massachusetts economy, most people don't know what it means to enter it. So we can help them learn that by building partnerships with government, academics, community, in industry. We all have to work together. That's the reality of the future. My goal is to integrate partnership into everything we do here at the university, including in the college I lead. We put together a network of local labs throughout the region who can help develop products, scale them. I mean, where do a lot of products go to die? Between research and development and commercialization and process development. We can help with that. We think about that all the time. And if any place should be able to do it, it's New England. Jeff gave me the 5-minute cue. By the way, shout out to Paige, who has to hold the cards for some really big personalities who are on the stage who don't take direction well, myself included.
You said you never say no.
I do never say no. So which is good because I'm essentially, I actually timed it. This is a miracle. I just wanted to end by mentioning or recapping what the biopharmaceutical analysis lab and bioinnovation is on the Burlington campus in Boston, just to give you an idea. We have a clinical diagnostic lab. By the way, we're open for business for any collaboration and partnership. I never say no. We think about biosecurity. Biomanufacturing is a lot of instrumentation that's shoved together, that's not made by the same manufacturer, and we disable lots of virus software and things like that to get them to talk to each other. Biosecurity, there's one major threat in the US a week that brings down a manufacturing line for a drug. We need to think more about that. So we do. We, of course, do analytics. I'm a mass spectrometrist. Actually, nowadays, I'm just a professional emailer. That's why many of you get emails from me. We think about biomanufacturing, single-use, integration, flexibility, all within the regulatory landscape. And then just lastly, before I end, the clinical lab has qPCR, digital PCR, next-generation sequencing. The whole entire floor can be made clinical. If I want to do clinical mass spec, I can. We have mock GMP to train apprentices from the Mass Bioed apprenticeship program. We have upstream and downstream, not enough downstream yet, processes. We can do protein chemistry, molecular biology, tissue culture. I have LCMS, I have NGS, I have flow cytometry, Flex. We have all the tools, and one of the core principles of what we do, where's Alex, that we do is bring in tools and put them through their paces, like I said, test them, generate data, publish papers, application notes, and things like that. And for the first time in my life, I actually have a team that can help me do this. Because I made Jeff take some other jobs so I could free some resources. No, just kidding. But it's really all about collaboration across the world, leveraging all of our campuses with our coalition for patients. At the end of the day, that's why I do what I do. Patients, patients, patients. And one of the newer projects we're doing is Nubian Square in Roxbury. They're gonna open a new building, and we're gonna mimic BATL in that building. I have $2.5 million for grants for equipment already. So once the space is done, we'll operate. The idea is that it's truly community-based, more people access to the life sciences. So with that, I will end by saying thank you to Barry personally for all of his mentorship and leadership to me over the last several years. The only one unfortunate thing is that I wish Barry was younger too, because we really started interacting after he retired, and not while I was one of the many colors on my ribbon at the Barnett. But he's still young. He's still got a lot of years. So I'll be on the phone with him for many. And to the Barnetts, thank you. And congratulations. I did? Oh, look at that. I'll take questions. Nobody's going to ask questions. That's good. Rick, what's your question?
What are you spending the 2.5 on?
What am I spending the 2.5 million on? Single-use bioreactors, mass spectrometer, flow cytometry, and then because the lab doesn't exist, I need the basics: biological safety cabinets, incubators, pipettes, all that stuff. Guess what? The bioscience people at Thermo like me. Other questions? I think Jonathan's concept of concept is awesome. We can't do what Jonathan can do. I would never say that. That's why we partner with him, and why he also has a visiting faculty appointment here, so we can do cool stuff. But the idea is that I can do some of those ideas I can do small scale in my lab, right? We can manufacture, like, if you think about AI and ML and protein design, right? Laurent might give me 10 molecules that he's designed, and I can manufacture them and characterize them. The idea of concept is a genius one, but we already knew that. Bring it here. Just rebuild it. Don't move it. We'll find you money. It's fine. Other questions? Just one more question. Barry, you don't have any questions? Jeff, you don't have any questions?
Jared, I'm extraordinarily proud of what you've accomplished. It's you have a shiny new building and done a lot. And I used to personally, this is just maybe it's a competitive nature, but I would pay attention to what Jared was doing in that facility, and then I looked at New Mexico, Colorado, Arizona, and giggle. We're still doing more than 8 states.
Exactly. It's true. It's true. Yes, we actually biobanked all of our COVID samples, so we can do much research for a long time. All right. Thank you. Oh, Ron, you get to come after me. Awesome.